ಇಲ್ಲಿ staff ಅವರು ಕೆಲಸ BEd student staff ಆ
अपना मोबाइल दूर रखो.
I request all the participants to kindly mute themselves. Good morning, all the participants. A heartily welcome to you all for the fourth session of International Webinar on Health and Nutrition Bite by Bite, organized by RKVY Raftar Agri-Business Incubator, College of Food Processing Technology and Bioenergy, Anand Agricultural University, Anand, to celebrate National Nutrition Month. I welcome Dr. Arif Sutar, Principal and Dean, College of Food Processing Technology and Bioenergy, Anand Agricultural University, Anand, all the departmental heads of college, resource person for today's session, Dr. Lakshmi Madam, and all the participants. I welcome you all. As a part of National Nutrition Month celebration, this webinar is organized by College of Food Processing Technology and Bioenergy, Anand Agricultural University, Anand. Awareness on healthy lifestyle is a reminder for everyone to live healthier lives. Good nutrition is one of the key to live a healthy life. Good and balanced diet improves the quality of life and helps prevent illness. And nowadays, people are becoming very curious today to make their diet healthy and fit, and also creating better attitude towards betterment for their health. With these words, I invite Dr. Videsh Pandey, Associate Professor and Head, Department of Food Processing Technology and Co-Project in Charge, RKVY Raftar Agri-Business Incubator, to brief about RKVY Rabi project. Sir.
Good morning.
Uh, Under this RKWare Avatar, we are running two programs, Pallav and Prasoon. Pallav is a program in which we are having a funding of around 5 lakh rupees, and Prasoon is a funding program under which a funding of around 25 lakh rupees it is being sanctioned. Currently, we have uh, incubated around 30 startups under these Pallav and Prasoon programs. And already we have organized a 5-day webinar on innovative agro food processing technologies for entrepreneurship development under this program. And this is second in the series to celebrate this nutrition month. Now I will be requesting Dr. Amiravani to introduce the speaker. Thank you.
Thank you very much, sir, for briefing about RKVY Rabi project. Now introducing resource person for today's session, Dr. Lakshmi Dave for today's webinar. Dr. Lakshmi Dave is a research officer in sustainable diets and bioactive peptides at the Redate Institute, Massey University, New Zealand. Her research focuses on the nutrient quality of dietary patterns and the sustainability of present-day food systems. Her research interests also include bioactive potentiality of dietary and gastrointestinal indigenous protein-derived peptides. Dr. Lakshmi is a strong advocate of women and the indigenous in STEM and leads the science communication activities of the Redate Institute. She received her PhD in nutritional sciences from Massey University, New Zealand, and has also worked as a research fellow at the TAGS Food Research Centre in Ireland. She is a registered nutritionalist at the Nutrition Society of New Zealand. Dr. Lakshmi is a proud alumna of SM Patel College of Home Science, Sardar Patel University, Vallabh Vidyanagar, and the Central Food Technology Research Institute, Mysore. With this word, I would like to invite her to address us on the topic food and gut indigenous protein-derived bioactive peptides, a new link in gut and overall health. Now over to you, madam.
Thank you so much for that introduction, madam. I would like to begin by saying good morning, all of you, and good afternoon and good evening to people who might be listening from other parts of the world. And I begin with a small greeting of the Maori people, who are the indigenous people of New Zealand, Aotearoa, New Zealand. Because I live in Aotearoa, New Zealand, I would like to begin by acknowledging them. Kia ora tēnā koutou katoa, which means hello everyone in Maori. And I'm delighted to be here. It's an absolute privilege to be able to talk to all of you and be a part of the webinar series of your college, FTPB. Especially in celebration of the Nutrition Month that's going on in India. As ma'am mentioned, I'm a research scientist at the Redate Institute, and I work both in the areas of sustainable diet and bioactive peptides. But I'll be talking to you today about bioactive peptides because it's a subject that is very close to my heart, and it's something that I'm very passionate about. In fact, it's also a topic I did my PhD in. So I hope you'll enjoy the talk, and thank you for coming. So the topic of my presentation today is food and gut endogenous protein-derived bioactive peptides, a new link in gut and overall health. So I've divided the contents of my talk today for you in 9 key parts, and I'll begin by talking to you about what the Redate Institute is all about, and then we'll move on to what are food proteins, which we've been learning about since probably all of us were in school, and then we'll move on to food protein-derived bioactive peptides, and then I'll introduce to you a quandary, a kind of a scientific puzzle that surrounds the area, concerns the area of, or the field of study of dietary bioactive peptides, that is the peptides that we get from our food. And then we'll talk about human gut endogenous proteins. I don't know how many of you might have heard about these group of proteins. I certainly hadn't when I was studying in school and college. So we'll look at what these interesting proteins are. And then I'll talk to you about what the research problem was. And then we'll move on to framing the objectives and then look at a tripartite or a 3-step methodology that we have followed to answer or answer the questions that we asked as a part of the objectives. I'll follow up with key results and conclusions from the 3 methods that we used, and then we'll end with the implications, the overarching larger implications or applications of our findings, and what kind of future work does this project or this topic entail. So this slide shows you where I am currently. And as you can see, it's a tiny island nation, New Zealand. It's really at the end of the world. There's nothing beyond that, beyond here, except probably Antarctica. So we are like 2.5, 3 hours away from Australia. And as you can see, we have 2 islands in New Zealand that make up New Zealand. So there's a North Island and there's a South Island. So New Zealand is actually probably as big as a mid-sized state in India. And the population is probably only as much as that of Ahmedabad, or perhaps even less than that. And the red dot shows you where exactly my institute is. So the town is called as Palmerston North, and it has striking similarities to Vallabh because it's an educational township, just like Vallabh. So it has floating population, mostly students and research organizations, and a big university called Massey University. And as you can see, it's a very small population. The town has a very small population. So the Reddit Institute is a center of research excellence that is hosted by Massey University. So the Reddit is actually a national partnership between 3 major universities of New Zealand, which is Massey University, the University of Auckland, and the University of Otago, along with 2 government research institutes known as Plant and Food Research and Egg Research. So plant and food and egg research are very much like the national labs that we have in India, like the ICMR laboratories or the CSIR laboratories like NIN and CFTRI and CCMB and so on and so forth. So apart from these founding collaborating members, this partnership, Reddit also works very closely with a lot of other universities spread across New Zealand and a lot of other research organizations and institutes as well. So I'll talk to you about my whānau. Whānau is another Maori word, which means family in Maori, and it's about the people who work in the institute. So we have scientific and technical staff, almost nearly 100, spread across the various institutes that I talked to you about, whom we collaborate with. And most of them are situated here in Palmerston North, but they're spread across the country. And we have a management staff, and then we are a hub, a training center, a major training center for future food science and nutrition research leaders, just like myself. And this is the Reddit management staff. So I'd like to highlight here Professor Singh, because I'm sure many of the students must have heard of Professor Harjinder Singh. You might have seen his textbooks, perhaps read his textbooks, or you will eventually get to see his textbooks. He's a world-renowned world leader in the area of milk protein chemistry, food structure, digestion, and just generally, he's one of the most well-recognized agricultural scientists as well from New Zealand. So, and then he is the director of the institute, and I now also work with Professor Warren McNabb, who happens to be the deputy director of the institute. So this map actually shows you that although New Zealand is really at the end of the world, we at the Reddit Institute are working very closely with world-class scientists across the globe. So there are many countries that we have collaborative projects going on in. So for example, in India, we have very close ties and work very closely with the National Dairy Research Institute, NDRI in Karnal. And we also work with Punjab Agricultural University, which is in Ludhiana. So what exactly is the science program or the vision of the institute, the Reddit Institute? So we work on future foods that are inspired by nature. So there's cutting edge science that happens in the institute. So how do we look at future foods? So we look at future foods from the perspective of food structures. So what are food structures? So food structures are every food, whether it is natural or processed food, food that we have made ourselves in a factory. All foods have a defined structure. So they have a macro structure and they have a microstructure, and the nutrients within the food are structured in a very specific way. The spatial arrangement is what is known as food structure, and that is what we study in detail. And we study this so that we can design foods that are similar to natural foods because they are naturally better for us. And then we have a food-human interface where we do a lot of studies in the next part of the core program. So in that, we look at how food structures, how different foods with different structures are digested in our body. So we use in vitro methods for this. So for example, we have a human gastric simulator that it looks like a giant box, but it's actually very good at mimicking how our stomach behaves. And that helps us understand how food breaks down in our body. And then we use in vivo methods, that is lab animals as well. So we use rats and pigs mostly. And also when we want to look at metabolic effects or any kind of a physiologically relevant effect, we also carry out clinical studies. So that brings me to my topic. So we begin with food proteins. So food proteins are not something new to any of us. We've been studying about food proteins since we were in school. We know proteins from foods are important. We know the role of dietary proteins. So they are macronutrients. Macronutrients in the sense that we require them as mammals, as humans, in relatively larger amounts than micronutrients. So for example, when we talk about protein as a macronutrient, I require, or an average human being requires, anywhere between 0.8 to 1 gram, or perhaps even more than 1 gram per kg body weight on a daily basis. So for example, if my weight is 50, then I require at least 50 grams of protein per day. Compare this to a micronutrient like iron. So iron, I might only require maybe around 30 to 50 milligrams per day. So why do we eat dietary proteins? So we don't actually eat dietary proteins for the proteins that we get or the polypeptides that we get out of those proteins. We actually eat the dietary protein for the essential amino acids that they give us. So we make use of the units that the dietary protein is made of, which is the amino acids. So essential amino acids are the ones that we require dietary protein for. So we don't require dietary protein for the non-essential ones per se, but for the essential ones. The essential ones are indispensable amino acids. That is that our body cannot synthesize them on its own. So therefore, we must get them from our diet for being able to grow, sustain, or to live. So this is so because we require dietary protein for these essential amino acids, dietary proteins are often referred to the only macronutrient that is required for sustenance or for life itself. So what happens when we eat proteins in our food? Like if I'm drinking a glass of milk, or if I've had a bowl of dal with rice, what happens to the protein? So like any other nutrient, any other food, it is subjected to digestion in our gastrointestinal tract or our digestive tract. So dietary protein actually breaks down or is digested by our digestive tract into its individual constituents or amino acids, as we spoke about earlier. So these amino acids are then the body decides that which are the ones that it needs, and then it carries out its regular metabolic activity. But research in the last few years, last few decades actually, has shown that amino acids are not the only end product of digestion of dietary proteins, which means that apart from amino acid, digestion of proteins can also lead to clusters of amino acids or peptide fragments, that is amino acid combined together, smaller than protein fragments, which are known as peptides. So these peptides could be anywhere between the range of 2 amino acids joined together. So it's a dipeptide, ranging up to maybe to pentapeptides, to maybe peptides as long as 40 to 50 amino acids. So it's very interesting to see that digestion of protein yields both amino acids and peptides. And some of these peptides may be resistant to further digestion along the tract, along the digestive tract. So what this means is that these peptides may not be digested further in the gut, in our stomach or small intestine. So what happens to these peptides? So some of them get absorbed, they go into the bloodstream, and they may get digested there. Or some of them actually completely remain resistant to digestion. So they remain as such. So they are peptide fragments as such. And they appear to have some kind of a regulatory mechanism. So these peptides that have a biological function, that appear to somehow tweak our cellular or tissue or any biological activity in our body, are known as bioactive peptides. And also, interestingly, bioactive peptides are not just produced during the digestion of dietary protein inside our gut. They are also produced when there is any kind of processing happening. So if there is any kind of a commercial enzyme that we are using, or if there is any kind of fermentation process that is being used in some product, it can generate bioactive peptides. And let's look at the historical perspective of bioactive peptides. When did anyone first encounter a peptide that appeared to have a regulatory activity? So the first known mention of dietary bioactive peptides can be traced back to 1950s. So in 1950s, when scientists were working with infants who had rickets, so children who were suffering with rickets. Rickets is a disease that is caused by the lack of vitamin D, deficiency of vitamin D. So children develop bow legs and other symptoms, bone-related symptoms. So what scientists observed was that when these children were, their diet contained one special kind of peptide, which is these casein phosphopeptides, the absorption of calcium appeared to increase. That is, the calcification of the bone ultimately increased in those children. So it positively modulated the physiology of the children, these peptides. And then later on, in the late 1970s, more milk-related peptides were starting to get discovered around that time. So during that time, some workers, Tishmaker et al. And others, found that bovine beta-casein, which is one of the major proteins in milk and dairy, it actually has a fragment which is released during digestion in our digestive tract, which behave like enkephalins. So what are enkephalins? Enkephalins, fascinatingly, are actually our own peptides. These are peptides that are naturally present in our body. We produce them ourselves, and they have one or the other kind of nervous system regulatory role. So they can be opioidergic. So it was a turning point for the field of bioactive peptides when it was discovered that proteins from dietary sources like milk protein, beta-casein, might have fragments of peptides in that protein sequence, which appears to be similar to the activity of the peptides that are naturally present in our body. Okay, so how are conventionally bioactive peptides defined in scientific literature? So they're defined as compounds that have a measurable effect at a physiologically relevant concentration. So there are two important concepts here. So the first one is they have to have a measurable biological effect. So what this means is that whatever bioactivity they're exerting, like if they're helping with calcium absorption, or they have some nervous system effect like we were talking about, then we should be scientifically able to quantify these effects using scientific analysis. And also, this should be happening at a physiologically relevant concentration. So for example, if we take the example of milk again, and if it's a beta-casein protein that is positively modulating someone's physiological function, and naturally after consuming milk, if one is able to generate maybe, let's say, around 2 milligrams or maybe 10 milligrams of beta-casein fragments in the digestive tract, then the effect should happen at that concentration or less than that concentration, and not double the concentration, because that will not be achievable in vivo, which is in our body. And secondly, when we talk about bioactive peptides, we largely only refer to peptides that positively regulate our physiological functions. That is, they have a beneficial role in our body. While strictly speaking, some of the peptides can also have negative effects, and these are also bioactive. For example, they can have some kind of toxicity or allergenicity or mutagenicity. So you might have heard the debate of A1 versus A2. So the A1 milk has a casein composition, amino acid composition that's slightly different, and fragments released from those A1 casein protein have been found to be implicated in various diseases. So those kind of peptides are also strictly speaking bioactive. But when we generally talk about dietary bioactive peptides, we are only referring to those ones that have a positive effect. So what are the kind of activities that these dietary bioactive peptides, what are the kind of activities or biological regulations that these peptides can carry out in our body? So bioactive peptides actually have a wide-ranging bioactivity in our body. Many of them can be multifunctional. That is the same peptide sequence. So suppose it is proline, proline, and valine. So that one sequence could have perhaps more than 1, 2, 3 activities. And also the range is so wide that in fact, there is perhaps no organ system in the body that cannot be positively modulated by dietary bioactive peptides. So bioactivity has been attributed to various different kinds of peptides across tissues and organs. So they can be anti-amnestic. Like I said, many of the milk proteins are opioidergic. Many peptides are anti-obesity. Many peptides are enzyme inhibitors. So for example, ACE1 inhibition. So that is an enzyme which when inhibited at the correct time can reduce hypertension. So there are peptides that can regulate heart health. There can be anti-cancer peptides, immunomodulatory peptides. There can be peptides that regulate the movement of the gut, hypocholesterolemic peptides, and peptides that help to bind minerals in the gut. So in general, there is a wide range of work, a wide range of effects that have been studied and that have been, and the dosages at which these peptides will exert these effects have also been found out. So now that we know about the incredible things that these peptides are able to do, I would like to request you to sit back and pause for a minute because I want to ask you 4 questions before we proceed. So my first question to you in relation to the quandary or the puzzle that I was talking about is that, what did you eat in your lunch yesterday? Try to think about it. The second question is, do you have the same breakfast for every single day, 365 days a year? My third question is, how is your best friend's diet? Is it exactly the same as yours, or how is perhaps your neighbor's diet or any other family member's diet? And my fourth question to you is, do you eat the same pulses throughout the year? Like if you eat tuer, then do you eat tuer 365 days a year, or perhaps you eat moong sometimes, or maybe urad dal sometimes? So various different kinds of sources of protein in your diet. So if you thought about the answer to these 4 questions, you would have come to 2 major conclusions. The first one is that our diet is not the same every day. In fact, it's not the same in every meal as well. So what I might have eaten in the breakfast is not the same thing that I'll eat in lunch or perhaps dinner. It changes. And then the other thing that we realize is that diets across people are not homogeneous. So we don't eat the same stuff. All of us don't eat the same stuff. Like I might perhaps be eating something entirely different to my neighbor or my best friend. In fact, I might be eating something entirely different to even the people I live with, my family members. So this brings me to this very fascinating world map of what protein intake looks like across various countries in the world. So there are some countries missing here that have not been mapped in terms of protein intake, but largely a lot of them have been looked at, scientifically analyzed. And what we find is, if you see the graph, the protein intake across countries and across populations and world regions varies between 47 grams per day to 118 grams per day. So that's a huge variation. So some of the countries that I was telling you that are not mapped here, and populations that don't have access to enough quality dietary protein, these are populations, these are cohorts that are perhaps eating only 7 grams of protein per day. So see the wide variation, see across populations and countries, there are some people who are eating just 7 gram, less than 10 gram of protein per day, and we don't know the quality of that protein, and ranging to the higher end where people are consuming more than 100 grams of protein per day. So like I mentioned before, our daily consumption of protein varies a lot, and there is data that shows this. And also the quality of protein varies. So for example, there are 2 people who are vegetarian. So one of them includes a sufficient amount of milk and dal and other protein sources in their diet, while the other person is perhaps averse to milk, does not like dairy products, and is very inconsistent with his or her intake of protein-containing, protein-rich foods. So that means that the essential amino acids that both these type of people will get will be different. So one might be getting significantly lesser amount of essential amino acids than the other. And then it also varies season to season. So some foods are available in some seasons, so we might not be perhaps eating them in other seasons. And this might be true for more true for some places in the world than the others, but seasonal variations have been noted in many places. And of course, it varies across populations, as we can see in this map, and also because cultural leanings and likings are strongly associated with the kind of protein that we eat. So let's look at the evolutionary. We saw the historical perspective. Let's look at the evolutionary perspective of food protein intake and bioactive peptides from food proteins. So pre-agricultural humans, those are our ancient ancestors, also known as the hunter-gatherers. So what did they eat? What was their diet like? So their diet was highly variable and a very, very seasonal diet. So there are many things now that we get throughout the year. So for example, we get armrests throughout the year now, but there was a time when our ancestors lived and walked the face of the earth when they did not have a consistent supply of anything at all. In fact, their diets were termed as feast and famine. So what does that mean? So for example, the hunter-gatherers, they lived in forests and very far-flung remote areas. So they go hunting, and they might today have found an animal and killed the animal and eaten it raw, or perhaps dropped it in the fire and eaten it. And the next day, when they were on their journey ahead, they perhaps didn't find even a single tree that bore fruits, or a bush that gave them any berries, or no animal to hunt. So for many days together, they might not have had sufficient food to eat. And what do you think they did what in terms of their milk and milk product consumption? So remember, this is a time when hunter-gatherers lived, pre-agricultural ancestors lived. There was no agriculture, there was no formal agriculture, there was no domestication of livestock. So they perhaps did not have milk and milk products in their diet like we do now. Definitely not as consistently as it's available to us, the supply now. So their milk consumption, they may have gone on to discover that they can consume milk of certain animals, but it would have been very, very sporadic, not a supply as consistent as our times. So millennia have passed since hunter-gatherers, and we are now anatomically modern humans, all of us. But what we must remember is that despite this huge evolutionary era that has passed, this time frame that has passed, our dietary nutrient requirements, or for that matter, our gastrointestinal system or digestive tract, is still very similar to our hunter-gatherer ancestors, to our pre-agricultural humans. So our human requirements and the way the anatomy of the gut functions is very similar to our ancestors. So just to quickly recap what we looked at so far, that is in terms of dietary protein and the supply of dietary protein and intake, we have seen that in countries or places, populations that have enough access to dietary proteins in their diet or in their food supply chain, the consumption varies anywhere between 40 to 100 gram, which is a big variation per day. And so the consumption varies daily. It varies in terms of quality. It varies from day to day across populations, seasons, and it also interestingly depends on the calorific intake of the diet. So the total calories that my daily diet contains will influence how the protein in my food is ultimately going to be digested. So this brings us to that quandary, the puzzle that I have been talking about. So the puzzle is that if our dietary intake of protein has been varying widely since pre-agricultural times, it has been a historically inconsistent supply of food protein in our food chain. And also, even though now we have ample supply, we don't consistently eat the same kind of proteins on a daily basis. How do you think our bodies might have evolved to develop the physiological functions that rely on dietary proteins or dietary protein-derived bioactive peptides alone? So in other words, like, the body is an extremely intelligent machinery. So if there is an inconsistent supply of something, the body will not make it the only fundamental source of regulation. So if dietary bioactive peptides are so important, but they are so variable, how did the body cope with this variation? Did it have any other mechanism to cope? So this brings us to these three questions. So to answer that quandary, we need to ask three more questions actually. So the first one is, are dietary protein the only source of protein in our gut, or is there another source of protein? The second question is, are dietary proteins the only proteins that get hydrolyzed or digested in our gut? The third question is, are dietary proteins the only proteins that give rise to bioactive peptides in our digestive tract? Answer to all these three questions is a big no. There is a hidden source of protein in our gut, in our digestive tract. Where does this protein come from, you might wonder? So from mouth through to the anus, as you can see here in this diagram of the digestive tract, we secrete a wide range of secretions into the gut, all mammals and humans, on a daily basis. So we have salivary secretions, we have gastric secretions, we have pancreatic biliary secretions, and all of these secretions contain hundreds of proteins, various different kinds of proteins, a diverse range of proteins. Collectively, all these proteins that we synthesize in our body and secrete into our gut collectively, these are known as gastrointestinal endogenous proteins or gut endogenous proteins, or in short, GEP. These proteins are essentially, their main function is to aid digestion and assimilation of the nutrients from our food. And if you look at the amount of GEP that is secreted on a daily basis, we secrete anywhere between 65 to 200 grams per day. So see this figure, if you remember the figure of what our dietary protein intake in that map looked like. So that was 40 to 100 and this is 65 to 200. So this is substantially more amount of protein. GP are secreted in substantially more amounts than dietary proteins. So this is a slide that puts together the two sources of protein that enter our gut on a daily basis. So the first one is obviously dietary proteins. So we eat anywhere between 40 to 100 grams. It goes into our gut, breaks down, gives us amino acids and bioactive peptides. On the other hand, we have a much more consistent supply of gut endogenous proteins. These are proteins that help in the digestion of dietary proteins, but they are themselves proteins. So what are some of the examples of GEP? So some of the examples of GEP are digestive enzymes like pepsin, trypsin, lipase, and so many other different enzymes that digest various different nutrients. And then there are digestive hormones like gastrin, somatostatin. Then there is mucins, there are lysozymes, there are other immunoglobulins. Then another key source of gut endogenous proteins is the protein in our epithelial cells. So the epithelial cells that line the gut. There's a lining inside the gut. The inner part of the gut is known as the lumen. So the lining around that inner part of the gut is known as the epithelial lining. This epithelial lining is turned over on a daily basis. So we know we have hydrochloric acid in the stomach. So this lining is turned over, slowed off. So it falls off and a new one is generated periodically on a daily basis. So when these cells are slowed off or broken off from the lining, they fall into the gut, they rupture, and the protein inside those cells, from those epithelial cells, is also released into the gut. Now that is a major source of gut endogenous proteins. In addition, we also have trillions of bacteria who actually make us a super organism. So we are not just humans, we are now known as super organisms in the sense that we are not just one organism. In our gut, we have trillions and trillions of bacteria. So when these bacteria die in our gut, especially a lot of them in the small intestine, they will also release all their protein, cellular protein content into the gut. And these proteins also get digested. And although bacterial protein are strictly speaking not endogenous proteins, they are not GEP, they're not gut endogenous protein because they are not our proteins, they are the bacteria's proteins. But they are also included in the estimates of gastrointestinal endogenous proteins because they are not dietary in origin. That is, they have come from the body in one way. So conventionally, proteinaceous compounds secreted into the gastrointestinal tract by specialized cells such as the salivary, gastric, hepatic, pancreatic, and other tissues are termed as gut endogenous proteins. And this definition also includes slowed off epithelial cells and bacterial cell protein in the gut. So what are the factors that can influence the secretion of endogenous proteins? So endogenous proteins secretion is firstly, majorly, one of the major things that triggers is the food that we eat. So depending on the food that you're eating, the quality of the food, the time you're eating, and the way your meals are spread across the day, these will decide how endogenous proteins are secreted on a daily basis. But some amount, basal amount of these endogenous proteins are always present in the body. So for example, if I'm on a fast, I'm not eating anything today, whole day, even then there will be some bare minimum basal amount of gastrointestinal endogenous proteins in the gut. So they are always consistently present. But when we are eating, what are the things in the diet that can influence them? So there can be various dietary and non-dietary factors. In the dietary factors, some of the key things are food dry matter intake. So which is the actual amount of nutrients in our food that we consume, can influence the secretion of endogenous proteins. Then the protein quality. So what kind of dietary protein am I eating? That protein, the dietary protein in turn, will influence the secretion of gastrointestinal endogenous proteins. Then dietary fiber. Am I eating soluble dietary fiber, insoluble, both? In what amounts, at what time? So that can also influence the flows of gut endogenous proteins. And then anti-nutritional factors in the diet, like phytate, tannins, can also influence what gastrointestinal proteins are secreted in what amounts. And then, of course, there is dietary electrolyte balance that can affect the flows. And let's talk about GEP. So dietary protein, we saw, once it enters the stomach, the digestion starts, and it gets further digested in small intestine, and ultimately it gets absorbed by the end of the small intestine. What happens to gut endogenous proteins? These are also proteins. So they are also subjected to the same gut environment, same conditions as the dietary proteins. So these gastrointestinal endogenous proteins also get digested. In fact, as far back as 1980s and 1970s, scientists did work on these proteins and found that nearly 80% of gut endogenous proteins are digested by the end of the terminal ileum, which is the last part of the small intestine. And this is very critical because that's where the absorption of amino acids happens. So a large part of the amino acids and peptides that would be released from GEP, just like dietary proteins, also get reabsorbed into the body. So this brings me to the objectives of this work. So the hypothesis of this work is mainly that what happens to gut endogenous proteins. So once they are secreted into the gut, they help to digest the dietary proteins, and then they help to digest the other nutrients in our food, but ultimately they themselves get digested. So when they generate amino acids and peptides, are there by chance any peptides there that might have biological function? So we frame the objectives in terms of 3 questions. So the first one is, do gut endogenous protein sequences contain encrypted bioactive peptide sequences? So what we mean here by encrypted is, do they have hidden bioactive peptide sequence in the protein sequence? So they haven't been released yet, but there is a protein defined, documented protein sequence of each of these gut endogenous proteins. Inside those protein sequences, are there encrypted, hidden, latent bioactive peptide sequences? Then the second question that we asked was, when this protein sequence with those hidden sequences of peptides, bioactive peptides, is subjected to digestion. So when gastrointestinal proteases and peptidases, that are enzymes that have the ability to break dietary protein and gut endogenous proteins, when they break gut endogenous proteins, do they release bioactive peptides in the gut? And the third question that we asked was that, does this process of the release of bioactive peptides happen on a normal basis during the course of normal digestion in vivo, in lab animals or humans? So we looked at answering these objectives using a 3-step methodology. So we began with in silico, which is bioinformatics based work. So we did a lot of mining. We extracted a lot of protein sequences and looked at what they look like, and saw if there were any encrypted sequences. And then we moved on to in vitro digestion, which include, after in vitro digestion, we got some peptides, and then we looked at whether those peptides have any bioactivity in vitro. And also we found sequences. We tried to characterize the sequences of those fractions which appeared to have some activity. Finally, we used lab animals for our in vivo digestion and tried to see if the same process actually happens in a animal. So before we move to the 3 methods that I was talking about and the results that we got obtained from each one of those methods, let's look at the gut first. So the gut is an amazing organ. Like every other part of the body, it is an extremely incredible piece of regulatory system, regulatory function. So what does a gut look like? So the gut is essentially a long hollow tube. So it is anywhere between 5 to 9 meters long, which is like a really long tube inside of us. And what is its surface area like? So its surface area, total surface area, is anywhere between 30 to 40 meters square, which is huge enough, large enough to be a standard apartment size. So just imagine how big the absorptive surface and the functioning surface of the gut system is. And I would like to ask you to think about, where is the gut situated? Where do you think our gut is? Like we feel when we talk about the digestive system, when we talk about, oh, I have a stomach ache, I keep my hand on my tummy. So we feel like it's situated within us. But think about the skin. Where is the skin situated? So skin is an organ, but the skin is situated outside of our body. So it is an organ, but it is outside our body, because it is in constant touch with our external environment. So it is an organ that is outside of our body. Similarly, the gut is an open-ended tube. So on both sides, it's open. It's in constant contact with the environment. So right through from the mouth to the anus, it is in contact with the pollutants in the environment, the air, the food that we get and put and ingest into the gut. So it's because it is in constant contact with the external environment, just like the skin, the gut is actually outside the body. It just appears to be inside the body, but anatomically speaking, the gut is situated outside our body. So when we looked at this fact that the gut is situated outside the body, we thought it was extremely important to classify bioactive peptides based on where they are situated in the body. So bioactive peptides broadly can be situated in body proper, which is actually inside the body. So organs such as the heart, the kidney, they're not in constant touch with the external environment. So they are truly, well and truly inside our body. So any peptides that you find there, they are in body proper. So what are these kind of peptides? So there are 3 types of these peptides, truly endogenous bioactive peptides. So they are actually truly endogenous. So biosynthetically derived peptides. So these have a signal peptide ahead of them when they are coded. And then when they have to exert their effect or do their primary function, then the signal peptide is cleaved and then they are able to do their work. So for example, serum albumin and so many other proteins. And then there are directly encoded peptides, bioactive peptides like open reading frame peptides, micropeptides. And then the third type of these truly endogenous bioactive peptides are kryptome protein-derived bioactive peptides. So these kryptome protein are like biosynthetically derived peptides. They do have a signal peptide. So they get coded, they get synthesized, but where the signal peptide is broken and then they carry out their primary function. But once they carry out their primary function, they are further broken down. Once their work is over, the body does not waste it. They are further broken down. And then they release bioactive peptide fragments in tissues. So for example, skin collagen. So skin collagen has a role. It functions to maintain the skin. But once the protein does its function, it is broken down by local proteases. And for example, there's a peptide fragment like endostatin, which is released from collagen, which is known to have a bioactive property. So the second broad category of classification of bioactive peptides would be outside body proper, which is the lumen of the gut. So lumen is the inner surface of the tube of the gastrointestinal tract. So any peptides that are there, that are generated there in the gut, are exogenous because the gut is outside the body. So any peptides that would be generated in that area would be external to the body. They are coming from external place to going and then eventually will go inside the body. So what could be these two type of, what are the two types of exogenous bioactive peptides? So the first one is obviously dietary protein-derived peptides. So we eat food, ingest it, and then peptides are generated in the gut. Similarly, gut endogenous proteins. Although gut endogenous protein has the word endogenous, endogenous means our own protein, one's own protein. But it is a protein that comes from the body cells and gets secreted into the gut tube. So because it is going into the gut tube, which is external to the body, any peptides that are generated as a result of the digestion of gut endogenous proteins are also exogenous to the body. So when we proposed this new classification, we did what most scientists do. We went ahead and published it. So what you see here is actually a snapshot of the classification review paper that we wrote about bioactive peptides. Okay, so why am I focusing on the gut? So in the earlier slides, I spoke to you about the fact that dietary bioactive peptides or any bioactive peptides can have a wide-ranging effect on almost every organ system of the body. So why then would I focus only on the gut? So there are two main reasons for this. So the first one is that once a peptide is released in the gut, like I eat, for example, I am consuming milk and I get a peptide out of milk protein in my gut when it is digested, then for it to actually cross the gut wall. So there is a brush border membrane that further digests these peptides, and then it has to cross the gut wall to go into the bloodstream. And suppose it has to have an effect in heart. So it has to travel through my circulatory system and reach the heart and exert a bioactive effect. But there are very few studies that have been able to find whether these peptides actually cross the gut border, do they go into the blood, and when they are in the blood, do the blood proteases further break them down or change them somehow, or when they reach other tissue, the target tissue, like for example, heart or kidney, will they be broken down or will they remain as such? And then will they be able to have an effect? So that is something that is still in nascent stages. So it's still being studied. So we don't know much about that part, which is called as the bioavailability of any compound. So in this case, bioavailability of dietary bioactive peptides. However, one thing is for sure, these peptides are definitely formed in our gut. So when protein is digested, what happens is that these peptides are released, and some of them are bioactive. So these bioactive peptides are likely to remain in the gut for a significant amount of time, because everything that passes through the gut has a transit time. So it stays in the gut for a certain period of time. Now, this certain period of time could be enough for that bioactive peptide to exert an effect in the gut, in that hollow tube in the lumen. It can exert an effect. How can it exert an effect? So that is because the gut is a complex organ system. So it's not just one thing that digests food and does nothing else. It has a lot of things that it houses within this complex organ system. So for example, there's a renin-angiotensin-aldosterone system. There's an antioxidant pro-oxidant balance system. There's an immune system within the gut, a huge immune system, very significant part of the immune system in the gut, which links back to the gut microbiota. The gut microbiota in turn regulates many functions in our body, ranging from our psychological makeup to our body weight, to how we feel about certain things, to genetics, to how we digest food, to a lot of things. And we also have an enteric nervous system in the gastrointestinal tract. So the gut microflora and the enteric nervous system in turn link or are associated to the gut-brain axis, which is also a very key modulatory bridge. And the enteric nervous system and the gut-brain axis links again to the central nervous system. So you see what is happening here is that if those bioactive peptides that are staying in the gut for some amount of time, which seems to be significant for those active molecules, they can go on and engage with receptors of any of these systems that are there in the gut, if they have that activity. And by that way, they will be able to modulate gut, and not just gut, but overall health of an individual. So this brings me to the actual experimental part of this work. So we will start with the in silico study. So like I mentioned, in silico is bioinformatics work. So we use softwares and tools and online databases to carry out this work. So it's also called as dry lab because we don't go to the actual lab, we work on computer. So it's referred to as dry lab. So what did we do in the in silico study? So we selected more than 20 gut endogenous proteins. So what you see here on the slide are just a few examples of some of the proteins, endogenous proteins that we chose. So for example, the secreted ones would be mucin, serum albumin, pepsin, other enzymes and some hormones and lysozyme, and keratin. So this comes from the epithelial cells that are slowed off, and also one protein of microbial origin in our gut. So once we selected these proteins, we needed to extract their amino acid sequence, their primary structure. So we went to a database, which is called as the UniProt KB. So this is where when a scientist discovers the amino acid sequence of a protein, this is where they enter it. So these are all documented, published data. So we go there. So the example that you're seeing here is of serum albumin, a gut endogenous protein. Significant gut endogenous protein. So we went to the website, we extracted the amino acid sequence of these proteins. So this is without the signal peptides. What you see in the purple box here is the sequence of serum albumin. So once we got the sequence, now we need to mimic or try to digest this protein using an online tool. So how do we do that? So we can take that protein sequence and enter it into an online tool, which is known as peptide cutter. So this tool allows us to choose enzymes that can digest our protein of interest. So if you see here, the circled ones, so I have chosen pepsin here to simulate gastric digestion. And then I've chosen chymotrypsin and trypsin to mimic or to simulate small intestinal digestion. That is the complete digestion of the protein of my interest. And then what we do is we enter, when we digest the protein, we get small peptide fragments. So we take those peptide fragments and then we enter it into yet another database that documents previously identified bioactive peptides. So for example, somebody might have identified a bioactive peptide from, let us say, urad dal, and they have gone and submitted it to the database. So if I have a new protein that nobody has studied and I digest it and I enter the sequence there, if it matches with something that was with urad dal, that was already found in urad dal, then it will tell me that it's a match. So this was previously found to be bioactive. So in this case, what you're seeing here is an antioxidative peptide, LK, which is leucine and lysine. So another thing that we can do is, like we found the protein sequence, instead of digesting it, we can enter the whole protein sequence into the database and see that in that big primary structure, amino acid structure of protein, where are the hidden bioactive peptides that already other researchers have identified, documented, and submitted to this database. So the highlighted ones that you see here in red and blue are all peptides that have been previously found to be bioactive in serum albumin, although I might have been the first person to look at serum albumin as a source of bioactive peptides. So apart from previously discovered bioactive peptides, we were also interested in seeing if proteins, gut endogenous proteins, have any novel sequences based on their structure that we predicted to have some kind of a bioactivity. So we found a lot of novel sequences. Then what we did was that the ones that we predicted to have activity, we went ahead and used microwave-assisted peptide synthesis. It's also known as chemical de novo synthesis. And then we purified them using reverse phase HPLC. And then we used mass spec, an instrument that helped us actually confirm the peptide sequence, the amino acid sequence of the peptide. And then we chemically synthesized and got powders of these synthetic peptides. So moving on to the results of the in silico study. So what did we find? So the first key thing we found was GP are a potential source of bioactive peptides. And that's how we published the paper, the snapshot of which you see here. So they have abundant bioactive peptides. Secondly, they have significant number of bioactive peptides that are released after simulated in silico gastrointestinal digestion of these endogenous proteins. And lastly, GP as a source of bioactive peptides are comparable. They can be compared as a source to dietary proteins. So it's not that dietary proteins are extremely rich in bioactive peptides and endogenous proteins are very few, negligible. No, they were statistically comparable. And then if you look at the first table here, so for example, beta-casein. So beta-casein is a known dietary source of bioactive peptides, correct? We'll focus on the first activity, which is ACE1 inhibition. So as I mentioned previously, inhibition of ACE at the correct time in patients who have hypertension can help reduce hypertension. So it is an anti-hypertensive activity. So beta-casein released after digestion, gastrointestinal digestion, 8 antihypertensive or ACE inhibitory peptides. Now look at serum albumin. Serum albumin is a gut endogenous protein. So serum albumin also released 17 ACE1 inhibiting peptides after gastrointestinal digestion. So it's very comparable to beta-casein, which was previously found to be a good source of antihypertensive peptides. Now, what happens in terms of predicted mean quantity of bioactive peptides released per day? Remember, I told you that concentrations have to be physiologically relevant. So we know bioactive peptides from milk can have a physiological effect from previous studies. So beta-casein in this case, for example, releases 87 milligrams of bioactive peptide per gram of beta-casein. Now look at serum albumin. Serum albumin, again, is comparable in terms of physiological concentration to beta-casein. So serum albumin was predicted to release 56 milligrams of bioactive peptide per gram of protein. So remember, we chemically synthesized peptides, we got those white powders. So what happened to those powders? So we actually got 19 novel peptides. That is, these are peptides that were never identified before. They're not there in the database. We were the first people to look at these peptides and their activity. So these peptides are novel. They come from gut endogenous proteins, not from dietary proteins. And these sequences had enzyme inhibiting activities, positive enzyme inhibiting activities, and antioxidant activity. And the IC50 of these peptides that we synthesized and their activities, IC50, that is the efficacy, how efficient they are physiologically, was similar of these novel peptides, GEP-derived novel peptides, to that of dietary proteins. And we also found that peptides with cysteine and proline residues showed maximum antioxidant and enzyme inhibiting activity. So we'll move on to in vitro digestion, which is digestion in the lab, so wet lab work and bioassays and characterization of peptides. So for the in vitro work, we chose 4 gut endogenous proteins. So trypsin, lysozyme, salivary mucin, serum albumin. So trypsin is from small intestine, a lysozyme is secreted throughout the gut, salivary mucin is secreted in the mouth, and serum albumin can also be found right from stomach till the small intestine. And we compared, for the purpose of comparison, we chose one dietary protein. So previously we chose beta-casein in silico. In this case, we have chosen chicken albumin because that is also known to be, it is a dietary protein known to release bioactive peptide in gut. So what did we do with these proteins? So these proteins, we took them to the lab. We carried out in vitro digestion, that is, we simulated gastrointestinal digestion using a standard consensus-based method called InfoGest. So InfoGest is a known method that is followed throughout labs that carry out this kind of digestion work. So we digested these proteins. So what would we get? We would get amino acids and peptides. So then we took these digested proteins, all 5 of them separately, and then we tried to fractionate them. So we tried to get peptides with lower molecular weight. That is, we focused on peptides with less than 3 kilodalton and 10 kilodalton molecular weight. And why did we do this? Because we know that smaller peptides have are known to have activity in the gut, bioactivity in the gut compared to the larger ones. And then we tested these fractions and the whole protein digest as well for a range of bioactivities that I've listed here, including ACE1 and antioxidant activity. And we use the standard microplate assays to test for all these activities. And finally, in the peptide samples, in the mixture of digests, where we found significant activity, we decided to characterize the peptides that may be potentially responsible for the bioactivity that we have seen. So what were the results that we saw here? So if you look at this STS page, it is an electrophoresis method. So what you're seeing here, the bands are actually protein bands. And this is a separation method, which you might be using in your labs. or might learn about soon. And the bands that you see here are separated based on their molecular weight. So these are proteins. So what we saw was that there is digestion happening. So we often use this method to see whether the protein is getting digested over time or not. So yes, the proteins are getting digested, which is a very interesting finding. Gut endogenous proteins are broken down into peptide fragments by gut proteases. And also what we see is that gut endogenous proteins were found to have significant ACE1 inhibitory and other activities, including antioxidant activities. Also, remember the fractions that we made, less than 10 and less than 3 kilodalton. Those fractions actually had much higher activity, as you can see in the graph here. The graph here shows DPPH inhibition, which is an antioxidant activity. So the lysozyme fraction, which was less than 10, had much more significant activity than the entire lysozyme digest. And also, we identified sequences of these bioactive peptides, as you can see here. The subsequences listed here. This is the final method that we use to confirm what we had found so far using in silico and in vitro methods. So we now move on to lab animal studies or in vivo studies. So what did we do in the in vivo studies? So we took 6 pigs. So we took the 6 pigs, we acclimatized them, got them adjusted to their new environment in the animal house, and then eventually we gave them test diet for 3 days. So what was the test diet? So the test diet that we gave to these pigs, 6 pigs, was protein-free. That is, in their diet, in their food, it was a normal healthy diet for growing pigs, which is recommended for growing pigs, but we removed all of the protein from that. So essentially, there was 0 protein that the pigs were eating for 3 days. And after that, we dissected out the stomach and the small intestine of the pigs to study the gut contents. So why did we feed the pigs protein-free diet? Because when we get the stomach and the small intestine sections out, when we dissect them out, whatever protein we get either in the stomach or the small intestine, it is the pig's own protein. So it is the pig's gut endogenous protein. There's no dietary protein there. So there's only body origin protein, pig's own proteins in there. And that was our, those are our proteins of interest. We want to know whether gut endogenous proteins in the pig, when normally the pig is eating, do they get digested? If they get digested, do they release bioactive peptides? So we purified the protein and peptide fractions from these samples, and then we tested them again for various bioactivities like we did in vitro. And then we moved on to characterizing peptides for finding which peptides might be potentially responsible for any activity that we are seeing. So this slide actually shows you the details of the sections that we've taken. So the stomach is one section. So the contents of the stomach. So when something is getting digested in the stomach, the animal has eaten something and it goes to the stomach and starts getting digested, and we section the stomach out. Then whatever we get inside the stomach, that content, digested content, is known as chyme. Similarly, in case of the small intestine, for the small intestine, we have taken the middle jejunum. So we didn't take the last part of the intestine where mostly absorption happens and we might lose some peptides. So we've taken the jejunal part, the mid part of the small intestine, and the contents of the small intestine are known as digesta. So what did we find? So it was extremely interesting. Again, an STS page here, which is doing the bands are protein and the separation is happening based on molecular weight. What you see here is that the see the lanes from, so the first lane and the last 2 are molecular weights, the standards. So in between are our samples. So the second, third, and fourth lane are stomach, and the rest are small intestine, the ones that follow after that. So you will see that in the stomach chyme, there is protein. There are bands, you can see them. But in the small intestine, there is a huge variety of protein. The diversity is much more than stomach. So this is in agreement with what was already known in literature. So we have again confirmed this fact that in the small intestine, a more diverse range of protein is likely to be secreted during digestion compared to small intestine. Now, it's interesting to see these proteins, but what is happening? Another thing that is key to what we were working on is that, are they getting digested? So yes, they are getting digested. Are they producing peptides, generating peptides? Yes, they are generating peptides. Do those peptides have activity? So yes, many of these peptides, when we purified them, got them out of the pig's stomach or small intestine and tested them for bioactivity, they had ACE1 inhibitory activity. That is, they could be antihypertensive or they could be antioxidant in nature, and several other activities as well. So we then went ahead and identified the bioactive peptide sequences from these samples. And what you see here, the subsequence, the top sequence, is actually a part of a previously identified bioactive peptide. So if you see, it fits somewhere in between in the lower one. The lower one is much longer. So the above, the sequence that you see on top is actually a subsequence, a part of that lower sequence. And that lower sequence has been established to be bioactive. So what we found was that these are the peptides that are likely to be causing the effects that we are seeing. So the effect that we are seeing, the activity that we are seeing, is clearly attributable to the peptides present in our sample. So what do all these results actually mean? The larger question is, what is the application of this work? What is the implication in terms of nutrition, in terms of our diet? So what the main thing that we found, if we look back at the results, is that dietary proteins, yes, they are an important source of bioactive peptide in the gut. However, they are an inconsistent supply of bioactive peptide in the gut by the virtue of the fact that we don't eat dietary proteins consistently. Secondly, just like dietary proteins give us bioactive peptide in the gut, gut endogenous proteins, our own proteins, have the ability to get digested and generate bioactive peptides in our gut. So what do we know about the diet? The diet can affect the dietary protein that we eat, and therefore it can affect the kind of bioactive peptide profiles that are generated in our body. But the diet also influences the gut endogenous flows. So in one of the slides, I spoke to you about how diet can influence the secretion of endogenous proteins. So if I'm eating more soluble fiber, I might have higher mucin secretion. So what does this link, this connection tell us? It means that it implies that actually we can use our diet to modulate not just the bioactive peptides that we get from our dietary proteins, but also the bioactive peptides that we get from our own proteins, that is the gut endogenous proteins. So it's a double whammy. So I can, if I'm, for example, suffering from suppose I'm suffering from, let's say, hypertension, and I want to reduce my blood pressure. I know that there are certain dietary proteins and certain endogenous proteins that have that can release bioactive peptides which can regulate my blood pressure. Then I can eat a diet that can help me generate those bioactive peptides in my gut. So it is a natural way of dealing with various non-communicable diseases. So the diet, dietary protein-derived bioactive peptides, and gut endogenous protein-derived bioactive peptides, all three together can modulate not just gut health, but overall health of an individual. So where to from here? So what more work needs to be done in this area? So actually, this piece of work is the pioneering piece of work in this area. There's perhaps no other group that is working on this in this field right now, in the field of gut endogenous proteins. But a lot of extensive work happens in RIDET on endogenous proteins, because we also look at these proteins in terms of their amino acid supply. And now we also look at them in terms of their bioactive peptide supply. So because this is a preliminary pioneering piece of work, a lot of work still needs to happen. So the first thing we need to do is we need to elucidate what exactly is the role of exogenous bioactive peptides in the gut. So what elucidating these the role of these peptides essentially means is that dietary protein peptides, bioactive peptides, and gut endogenous protein derived bioactive peptides in the gut, are they together forming some kind of a regulatory system? Is there a feedback mechanism? Is there a peptidergic regulation that is happening in the gut, which is hidden and we had no idea about it so far and are only beginning to see? And then the second thing we need to find is that gut microbiota. Gut microbiota is a huge source of endogenous protein. How are they influencing the generation of bioactive peptides from the gut endogenous proteins? So microbiota changes rapidly on a daily basis, and it can be positively, negatively modulated through our diet. So when we do that, when we eat the wrong kind of things or the right kind of things, what happens to the supply of bioactive peptide in the gut? The third thing we need to look at is developing a consensus-based method to distinguish between dietary and gut endogenous derived bioactive peptides. So why do we need to distinguish? So if I've eaten, for example, a dietary protein, like let's assume that I ate some dal, and it released peptides in the gut which have bioactivity. And my own endogenous proteins also released a peptide which is similar to that peptide, same sequence to dietary protein peptide, and that also has an activity. So how am I going to distinguish where this protein came from? What was the origin of the protein? Did it come from the dal that I ate, or did it come from my own protein? So we need to be able to distinguish between the two for us to be able to understand how the exogenous peptides work in synergy. After that, we will probably also need to look at the bioavailability of exogenous peptides. So this is an area in which a lot of people are working across the globe. So bioavailability, like I mentioned, is a generation of the peptide in the gut, and then it crosses the gut wall and goes to various target tissues through the circulatory system by traveling in our blood vessels. So how does that happen? So when it enters the portal circulation, does it remain as it is? Does it actually reach the tissue? Is it able to exert an effect? We need to figure out the bioavailability of those molecules and compounds. And finally, we will need to identify the gut bioactive peptide pools of healthy individuals. So it is very likely that because these peptides have positive regulatory functions, the bioactive peptides pool or the profile of a healthy individual is likely to be different to that of an unhealthy individual. So then when we know the differences between healthy and unhealthy individuals, we can use these pools, these bioactive peptide pools in the gut, as a biomarker to probably identify disease before it sets in. And if it has set in, how can we change these pools to ameliorate the disease? That brings me to the acknowledgements. Very grateful to my institute and also to Chagas Food Research Center in Dublin, Ireland. And these are my email ID and my Twitter handle. I'm very happy to answer any questions that you might have, and please keep in touch.
নহয় নহয় এতিয়া কিবা ডায়েটৰী প্ৰটিন কিবা এটা ৰবা গাত
Thank you, madam,
প্ৰটিন বুলি এটা
for
ওলাইছিলে
your nice
মানে
presentation.
ৰেজিষ্টাৰ কৰিব নাই সেইটো শুনিছিলোঁ
I thank you for accepting our invitation within such a short notice.
Thank you.
Now I will be requesting Dr. Modi to take over the session for question answer. You have elaborated about this food peptides and biopeptides, and specifically their role in gut, and probably some of the biopeptide they might be beneficial for the gut in terms of that they might be promoting the growth of probiotics for the benefit of the health of the human being. Now I will be requesting Dr. Modi for this question answer session. Thank you, madam.
Thank you.
Yeah, thank you, sir. And thank you very much, ma'am, for the wonderful lecture. It was really very much knowledge sharing lecture. And now I am requesting the audience to please, one by one, they can unmute the mic and ask the questions to the speaker.
Hello.
Yes, please, sir.
Yeah, thank you very much for very informative and well explained lecture, ma'am. My question is, endogenous protein, if it is exclusively by human tissue cells, then how can we discriminate whether this particular protein is produced by human tissue cells or gut microflora?
Yes, thank you for the question. That is a very interesting question. What happens is a large part of the endogenous proteins already have been sequenced. So when we look at the human proteome project, we already know, like, we have 80,000 to 90,000 peptides, and each one of them have been have been characterized, the protein sequences have been characterized in great detail. So we know the sequence of each of these proteins, and but we don't know the sequences of all of the trillions of microbiota that are there in our gut. So there can be overlaps. In fact, there can be overlap with dietary protein as well, like I was mentioning. So, so the bacteria in our gut can have a particular small protein that has a sequence that can match the sequence of the endogenous protein, a peptide inside an endogenous protein, that can in turn match the sequence of a peptide in a dietary protein. So unless we label these proteins, so there are isotope methods to label these proteins. So in some studies, for example, when we are doing dietary protein studies, if we feed the animal protein, in my case, in this study, we didn't feed the animal protein, but we do studies wherein we feed the animal protein. So when we feed a dietary source, we can label it using isotopic methods. And then when the animal eats it, and then when we analyze the sequences, the one that is obviously labeled is coming from diet, and the ones that are not labeled are coming from the animal itself. But within the animal itself, whether it is coming from an endogenous protein of the animal or the bacteria, it might be difficult to find out, especially if it's a very small peptide, because small peptides are very difficult to characterize using mass spec.
Okay, thank you, ma'am.
Thank you, ma'am. Now, Dr. Ruma Bhattacharya, you wanted to ask something. Please, ma'am.
Yes. First, congratulations, Dr. Lakshmi Dave. Very nice presentation.
Thank you so much.
Oh, so much well explained, and I'm very much interested in protein quality, etc. I am a professor here in Assam Agricultural University, but it was really thought-provoking about gut endogenous protein. So my question is, because we speak about gut microbiome, the environment inside a gut microbiome. So I think there will be a relationship between gut microbiome condition and endogenous protein. Can you put some light on it very simply? Very simply, that's my query only. Gut microbiome, environment.
Sorry, I didn't hear the last part of your question. Sorry, ma'am.
Gut microbiome, environment, yes, and the production and utility of gut endogenous protein.
Yes.
Like, is it a factor? Is it a factor?
Yes, it is very much a factor. So in previous studies, so before we did this study, there were other studies happening in our institute, and yes, they were done both in rats and pigs. So what we found was that in the gut endogenous proteins, the various proteins that I mentioned, yes, we also include bacterial proteins in that definition. So okay, you would be surprised to know that 45% of gut endogenous, yes, proteins actually come, yes, from the microbiota. So they're not ours, but we count them as ours because they the microbes live in us. So that 45% is like nearly half of the composition of the gut endogenous proteins. And that composition is highly malleable. It changes throughout the day, and it changes rapidly. Yeah. So it is actually one of the modes that is used for treating non-communicable diseases. So if someone's eating a wrong diet, simply by changing their diet without any medication, they can modulate gut microbiota to change it to favorable microbiota, and therefore reverse the disease condition. So because they are so malleable, they have a huge impact on the endogenous protein. And then because they have a huge impact on the endogenous protein, the peptide that will come from those proteins will also be affected.
Okay, thank you. Another query, very simple query. It's like throughout the life cycle, the vulnerable groups have different type requirement, protein requirements. Okay, recommended dietary allowances are different because the conditions are different. But at the same time, whether the genetic potential of that particular person or particular target group, their nutritional requirement and GEP has some kind of impact, because genetically people are different, and moreover during very vulnerable conditions like pregnancy or infancy, requirement is more. That time, whether GEP isn't like an what to say protector, or it is really very important to think about GAP at that particular moment.
Yes. Thank you for the question. It's a very interesting question. It is actually, GP is actually likely to have a very significant role in terms of protein requirements of humans at various life stages, and different protein requirements depending on their lifestyle and other factors, or any disease that they might be suffering. This is because when we look at protein requirements of human in terms of dietary proteins, so the dietary protein requirements in human nutrition, we also actually assess the endogenous flows in the gut. So the new method for assessing the protein quality of proteins from our food, dietary protein quality, is known as DIAS, the digestible indispensable amino acid score. So this score actually takes into account the gut endogenous amino acid contribution as well. So what we feel physiologically might be happening is that the gut endogenous proteins act like buffers. And like you rightly pointed out, there is the angle of genetic predisposition, genetic variations, and also as we age, for example, our gut kind of shrinks like every other organ in our body. So it's not able to function to its maximum capacity. So our diet, we need to change our diet accordingly. So when we change our diet accordingly, we also, as a result of that, modulate the endogenous protein flows. And that in turn acts as a buffer for us to be able to cope with these variations. So like when I was talking about the map where we see protein intake varies across countries. So different people are eating different amounts of protein, but largely everyone who is getting sufficient protein or even slightly less protein seems to be doing okay. That is because these endogenous proteins have a reactionary effect to what we are eating. So if I'm not eating enough protein, we have seen in animal studies that my endogenous flows will increase, but they will only increase till a point and then plateau. So the body is able to cope to a certain extent. So it acts like a buffering mechanism to be able to cope with external and genetic variations.
Thank you so much. And this is really innovative. And I have taken your email and Twitter handle. So I may be in contact. I am a professor. I'll be retiring by another 1 year. But still, I'm very much interested. And I can at least suggest some of my students who all are working on protein.
That would be wonderful, ma'am. Yeah, that would be wonderful. We are very happy to, thank you, ma'am. We are very happy to talk to any students who might want to work here or set up a collaboration with you in your lab. We really look forward to it. Thank you so much.
Thank you. Thank you so much. Nice meeting you. Thank you.
Hello.
Yes, please, sir. Ask your question.
Hello. First of all, congratulations for Lakshmi Devi, ma'am, for this nice talk and elaborative lecture. So, this is Dr. Ajay Desai. I have been 3 years in New Zealand in the Lincoln University, working with Dr. Charles.
Wow, small world.
Yeah, it is working with Charles Brennan. You must have heard about this. Yeah.
Yes, I think I have. Yeah.
I am the member of the Reddit Institute. That's I got the placement award. That's you.
That's brilliant.
Yeah. So nice talk. One question is regarding, because I am from the fisheries background and I did my PhD in fisheries. So what your studies conducted is specific age group for this, because the digestion property of the different you can say the age group is different, because enzymes, because when protein we have taken the proteins, so certain enzymes, for example trypsin, chymotrypsins, enzymes are there, they have capacity to digest. So maybe their effects on certain we can age groups. First question. And second question is in seafood, because seafood you can say have the high digestible proteins. So any difference we have found it's on seafood proteins or in milk proteins?
Yes, thank you for the question. No, we weren't looking at any age group, unfortunately, in this piece of work, because this was just to show, this was the first conceptual proof for us to understand whether like dietary proteins, endogenous proteins are able to generate any bioactive peptides during the course of normal digestion. So that is the reason we used pigs in our study, and they were growing pigs So it was that age group, so 6 months, around 6 months of age. So what we see, the peptides generated there are, like you mentioned, likely to be different to the adult pigs, and it's the same with humans as well. So it's going to be different across age groups and across sex and across perhaps dietary habits and a lot of other factors as well. So the reason we didn't look at age was we were trying to look at the fate. We were trying to find the fate of endogenous proteins. We know they have primary function. We know that they go into the gut and digest the food that we eat. But then what happens to them after that is what we were trying to find. So we will definitely be looking at more detailed work in this area in terms of like how does it affect different people, different age groups, and perhaps even different ethnicities, which is likely to be different, just like the gut microbiota. And also your second question regarding proteins that are very well digested and absorbed. So yes, we do a bit of work in that area. So for example, beta casein, a slow releasing protein, but then completely digested, completely contrasting to beta lactoglobulin. It would be very for many people it's not a protein that is very easily digested. So these proteins can and the peptides that they ultimately generate, the resistant peptides will be different in these proteins. So that in turn is likely to have an effect on in terms of what is the bioactive peptide pool that we see in the gut.
Okay, thank you. Thanks, ma'am. Thanks.
Thank you, ma'am. And one more question I have received from Sri Dilip Singh Jhala, and he is asking that in Gujarat, majority of the people are eating tuver dal daily. So, is there any harmful effect or bad effect of this?
No, not at all. Because traditionally, if you look at our diets are extremely intelligent. The Indian traditional diets and recipes, because there is a defined cooking process. So so we don't eat dal like probably it would be consumed in the Western world. So what our aim when we cook dal is to completely soften it. So we use a pressure cooker in general. We even, in fact, soak it for quite a long time. So the nutrients within those dal proteins are likely to be assimilated better because of the traditional cooking practices. Also, if you look at food combinations in traditional Indian diets, so dal, roti, dal and rice, khichdi, and these are all standard combinations. So these are all combinations that are thought of or have been there for thousands of years because it is known that they complement the lacking amino acids, and no, it's a perfectly healthy diet to have.
Okay, thank you, ma'am. And if somebody from audience want to ask the question, please. Mr. Nayak. Okay, one question is posted in the chat box. I am repeating it. Don't we lose the minerals and or water soluble vitamins by soaking?
It depends on what we are soaking. So so micronutrient is something that we get majorly from fruits and vegetables. So nobody would like soak cut a fruit and a vegetable and soak it for too very long or discard that water, which is what we encourage people to do. So if you are using water, reuse that water for something else, or don't discard that water in case of fruits and vegetables. But in case of dal, what we do is, or in case of rice, or when we are making a khichdi, when we mix rice and dal, what we essentially do is we wash it thrice to completely get rid of any debris or any dirt that might be there. And then we soak it in clean water. And then we use the same water to cook that generally. So we aren't actually losing anything much. Yes, of course, there is a little bit of micronutrient loss that is going to happen, which is unavoidable by cooking. Some of the heat labile ones might go, and some of them might perhaps change. For example, some proteins might denature when we cook, or there might be some mineral protein interaction that might happen. But then largely, most of the things that we soak, we don't generally recommend discarding that water because you would have soaked it in clean water. especially cereal grains and legumes and things like that.
Hello, madam. One question is like lactose sucrose intolerance. Does protein intolerance may happen in human body?
Protein intolerance?
Like lactose sucrose intolerance in human body, does protein intolerance may happen in our body or not? Please
Yes,
clarify.
yes, of course. So just like lactose intolerance from milk proteins also, some people are intolerant to them, or they might react um, uh, unlike normal people. So it might not be clearly be able to be defined medically as intolerance, but some people might be able to digest milk proteins better, especially when we talk about A1 versus A2 proteins. So A1 is the evil protein, so-called evil protein, while A2 is the protein that we find in the beta casein that we find in the traditional breeds of our Indian cows. So that has lower disease causing propensity or perhaps even intolerance than A1. So definitely, in fact, a lot of allergies that you see are owing to protein. So for example, several people are allergic to eggs. Some people are allergic to groundnuts. So when they eat sing, khari sing, their face swells up and they have a reaction. So that is because of the protein in those foods. So you're absolutely right. So just like any kind of a sugar intolerance, we can have protein intolerance as well, or allergenicity as we would call it.
Thank you.
Thank you.
Thank you, ma'am. So I think all the questions you have answered very well, and
Thank you.
Audience is very much satisfied with the lecture and the knowledge you have shared. So I am handing over the mic to Dr. Amir Rawani for the validatory session. Thank you, ma'am.
Thank you.
Thank you very much, Lakshmi Madam. It was really very much informative lecture on the bioactive peptides.
Thank you.
Thank you very much. Thank you all the participants for your active participation during the lecture. Thank you all the participants and YouTube viewers also. There is announcement for tomorrow's lecture. Tomorrow will start at 5 p.m. in the evening, and the lecture is on advanced food technologies for healthy nutrition and industry perspective by Mr. Ashish Anand. He is Senior Director, Global Beverages, R&D, PepsiCo, New York. So tomorrow we'll meet again at 5 p.m. on 25th September. Okay, thank you all. We are just signing out and we'll meet tomorrow. Thank you.
Thank you so much. Thank you so much for having me. Thank you, everyone. Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you so much.